Hello, everybody. Welcome on my talk. My name is Eva Lusig from INOC. We are based in Germany, and we help to the transition to a more sustainable agriculture by developing biofertilizers based on arbuscular mycorrhizal fungi, even if many challenges remain for the routine use of these products in agriculture. So as a result of geopolitical and climate crisis, the conditions for food production have become more complex during the last years. We see an increase in the price of fertilizers, which are also less available, and record heat and drought episodes regularly affect plant growth and yields, at least here in Germany. And to make the situation even worse, the conventional agrochemical-based agriculture damaged the soil ecosystem functioning in many fields, like practices as application of high doses of fertilizers, fungicides, and pesticides, or intensive tillage, were all detrimental to plant beneficial microbe diversity and activity. So we really need now to restore our soils and go towards alternative cultivation practices. And the regulations in Europe also restrict more and more the use of phytosanitary products. And plant growth promoting microorganisms may represent one of the solutions. Arbuscular mycorrhizal fungi form symbiosis with more than 70% of terrestrial plants and constitute a key functional group of soil biota. And they can not only improve plant growth through increased uptake of nutrients, but have also non-nutritional effects by alleviating biotic or abiotic stresses in plants and in stabilizing soil aggregates and microbial communities. During the last years at INOC, we have developed a successful commercial product called Advantage, which is a concentrated inoculum based on colonized root fragments. But this product had absolutely to be optimized for its use in agriculture because the low volumes to apply and the powder-based composition represented huge bottlenecks for field application. And within the master project, we were able to develop clay-based pellets, which fulfilled the most important criteria of a formulation, which are to protect the fungal propagules from the soil influence, to concentrate locally the propagules, help to control the dosage of the product while being easily applicable with existing machinery, and of course safe for the environment and presenting no inhibitory effects on fungal or plant growth. And after field validation on sunflower and corn, where really good results were obtained, as well on yield and drought stress alleviation, the product is now available on the market, and we hope in the future to test and adapt it as widely as possible to different crops and soils. Thank you.
My name is Ronnie Groben. I'm a project manager in the microbiology group at Martis, and I will give you a short presentation of the microbiome in aquaculture innovation. With a growing global population and the need for healthy, nutritious food, seafood is in demand as never before. However, current fishing practices put a lot of pressure on marine ecosystem, and the fish stocks worldwide are often in danger of overfishing, making them unsustainable in the long term. Aquaculture, which is the rearing of fish or other aquatic organisms in large tanks on land or in sea pens in the ocean or in lakes, has the potential to solve the problem and produce high quality food in large amounts. As a result, aquaculture became quickly the fastest growing industry in the food sector. There are, though, problems associated with growing aquatic organisms in aquaculture, and one of them is the potential occurrence of pathogens among the high-density population of animals. A routine monitoring system for aquaculture pathogens is therefore necessary that gives the fish farmers fast and reliable information about if, which, and how many pathogens occur, and if measures need to be taken to protect the health of the fish stock and, of course, of the consumers. Current methodologies, like, for example, cultivation methods or immunological diagnostics, are normally focusing on only specific pathogens. They often take days or weeks and normally require specialized laboratories and highly trained staff. The Oxford Nanopore sequencing system that was tested as a monitoring tool in the master project has the potential to overcome these obstacles. In master, we developed a rapid protocol for pathogen detection and microbiome screening that allows for the early detection and prevention of diseases in the aquaculture industry. Through trials, adjustments, and improvements of the sample preparation method, which is a crucial part of the detection system, we were able to obtain high-quality DNA that could be successfully sequenced by the Nanopore system. The advantages of the new monitoring systems are manifold. It is capable of identifying all microorganisms together, so it's not only focusing on one or two potential pathogens at a time. It works with a wide range of aquaculture-related sample types, like water samples, fish tissues, or, for example, the biofilters from the tanks. It does not need sophisticated laboratories and can be done on-site at the aquaculture facilities, so that there is no need to transport them to dedicated laboratories and waste time. The method is easy to use, and staff can quickly learn it. And last but not least, the whole protocol will only take a few hours until the first data is available, compared to days or weeks with traditional methods. This innovation in this new protocol was developed within the master project by researchers from Martis, Nanopore, and Lífsgleði, and we would like to thank all partners for the fruitful collaboration.
Good afternoon, everybody. My name is Stuart Kerwin, and I was part of Work Package 3. And today I'm going to talk about breeding lower methane emitting cattle. So the objective within Master was to investigate the role of diet, host genetics, feed efficiency, and rumen microbiome on the environmental output in beef cattle. And one of those tasks was to develop an estimated breeding value for methane. So we know that methane is heritable and that we can select for it. And it's permanent, it's cumulative. So whatever we select for now, it has implications for future generations of livestock. Also, in relation to selecting methane, it's suited to situations in livestock systems where it's not suited for them, or basically that they can't implement feeding of dietary supplements that mitigate against methane. So we know that methane is linked to dry matter intake, so we need to be careful on how we select for methane, and that whatever we select for, that it doesn't have any negative effect on performance. So over the course of 2 to 3 years, we collected a large, I suppose, a large data set on animals. There was over 1,500, and I suppose methane data collected on over 1,500 animals, and then there was also performance data that was linked to over 3,000 in relation to feed intakes and their performance, and that was combined to give us the first, I suppose, the first genetic proofs for methane. So essentially, what that means is that we can now actively select sires for reduced methane output. Just to acknowledge some of the people that worked on this work package: Sinead Waters, David Kenny, and Paul Smith from Teagasc. But also I'd like to acknowledge Stephen Conroy from ICBF, who facilitated a lot of this work too.
Good afternoon to everyone. My name is Abelino Álvarez Ordóñez from University of León in Spain. And today I am going to talk to you about a very relevant innovation of our project Master, a protocol that we have validated for the mapping of microbiomes in food industries. The characterization of microbial communities that colonize food industries is extremely important, as these communities can have an enormous impact on the quality and safety of the food that we consume. Monitoring and surveillance of microbial contaminants has been traditionally approached through the cultivation in laboratory media and the detection and enumeration in culture plates. However, these culture-dependent approaches are time-consuming and only allow to analyze one microorganism at a time. In recent years, the huge developments in DNA sequencing technologies have granted the possibility of approaching the characterization of microbial communities without the need for cultivation, which offers many advantages as information on multiple microorganisms can be simultaneously obtained. However, food industry samples are challenging as they are expected to contain low microbial concentrations and may have residues of cleaning agents and organic materials. In addition, there are no protocols available in the market for the isolation of DNA of high quality from this type of samples. In MASTER, we have developed a complete protocol with this aim, which with the standard operation procedures for all the steps of the microbiome analysis, including the sampling in the facilities, the sample manipulation in the lab, the extraction of the DNA sequencing, and the final computational analysis. Remarkably, our project partner QIAGEN, a leader in the market of DNA extraction kits, has collaborated with the development of a new DNA extraction protocol adapted to the special needs of our food industry samples. The new protocol was successfully validated in more than 2,000 samples from food processing environments, from raw materials, end products, and even from operators taken in 114 food processing industries of different production sectors from 5 different European countries. The development of this innovative protocol, which we hope will have a very significant impact for food industry operators and also for analytic laboratories, has been possible thanks to the collaboration of different partners of the MASTER project, including QIAGEN, the University of Naples in Italy, Chagas in Ireland, FOXI in Austria, CSIC in Spain, MATIS in Iceland, the University of Trento in Italy, and our University of León in Spain. To finalize, I would like to thank all the food business operators that allowed us to test our protocol in their facilities, and I would also like to thank you for your attention. Thank you very much, and good afternoon.
Good afternoon, everybody. My name is Laura Nine, and I'm a postdoctoral researcher in University College Cork. And I want to give you a brief overview of the work that we have carried out during the MASTER project. And our task was the upcycling of brewing byproducts to produce functional food ingredients. So we were working mainly with 2 brewing byproducts. So the first is barley rootlets, which are obtained from the barley malting process. And the second is brewer's spent grain. Brewer's spent grain is the most abundant byproduct of the brewing process, with about 36 million tons of this produced globally each year. Currently, these byproducts are used as animal feed and sometimes disposed of in landfills, which is quite unsustainable. However, they represent a really valuable source of nutrition, containing about 30 to 45% dietary fiber and between 30 and 40% protein. So to date, the addition of these byproducts into food as a nutrition source has been challenging due to the negative effects that they can have on food quality attributes, such as flavor, texture, appearance, and overall acceptance. However, one method which can improve the performance of these types of byproducts in foods is fermentation. So in our research, we have seen that the use of fermented ingredients in foods, they have a better performance in food, they can enhance the nutritional aspect of a food, they can improve the sensory attributes of a food, they can extend shelf life, and they can also provide health benefits to the consumer. So in our task during the MASTER project, we used fermentation technology to upcycle brewing waste materials into functional food ingredients. So we developed a range of different fermentation protocols, and we tested various different lactic acid bacteria, each which had their own unique characteristics. And with this, we were able to produce a range of functional fermented ingredients, which can be applied in a variety of different food matrices. So we looked at it firstly in an application in bread, and what we saw was that interestingly, each ingredient had a different function in the bread. So they all improved the fiber content of the bread. Some improved the volume of the bread and gave a softer bread with less hardness. Others increased the shelf life of the bread, and some gave a really nice sourdough bread taste profile. So what we concluded is that we can use fermentation to produce innovative upcycled food ingredients which have a better performance in foods and can be applied in a range of different foods. And what was really interesting was that we can tailor the fermentation in order to produce ingredients which all have different functions depending on what kind of food and beverage you want to apply it in at a later stage. So thank you very much for listening to this short presentation. I would like to thank everyone in UCC, which has worked on this project for the last 4.5 years, and also our collaborators in AB InBev for providing the raw materials and also for their scientific input during the project. And I'll be happy to answer any questions you have. Thank you very much.
Hello, everyone. My name is Fernanda Godoy Santos. I'm an animal scientist and a doctor in agricultural microbiology. Currently, I work at Queen's University Belfast. And today, I'm going to provide you with a brief overview of our work in the Master Project, where our group of researchers are creating new bacterial mock communities. So, as you have seen in previous presentations, microorganisms play a crucial role in agriculture, human health, and in the environment. To better understand bacterial dynamics, interactions, and functions, scientists have created mock communities. These artificial populations mimic the natural population, serving as simplified models for studying bacterial behavior in a controlled environment. In our bodies, microorganisms are everywhere, and their population diversity is influenced by the environmental conditions. Here, we have two examples, a median plate showing aerobic microorganisms from the skin, and another median plate showing anaerobic microorganisms from the intestine. Looking at these pictures, you can clearly observe the difference between the two populations. In addition to their role in understanding bacterial dynamics, interactions, and functions, mock communities have several potential applications. First, they are important in ensure representativity. As we mentioned earlier, microbial population can vary depending on the environmental conditions. Therefore, scientists carefully select specific bacteria to create mock communities that represent different types found in the natural settings. Mock community also play a crucial role in quality control and innovation. They allow researchers to predict and assess bioprocess performances, and also offer exciting possibilities in food product development and innovation. By manipulating the composition of the mock community, scientists can optimize the characteristics of the final product. This can lead to the creation of new flavors, textures, and even improved nutritional profiles in food. Another significant benefit of the mock community is their contribution to the research reproducibility. Scientists can compare and evaluate different techniques using mock communities, ensuring reliable and consistent results that can be replicated across experiments and studies. In this context, the scientists involved in the Master Project focus on creating two bacterial mock communities, one aerobic mock community food-associated, and one anaerobic mock community rumen-associated. Throughout the project development, new bacterial cultures were isolated, characterized, and carefully selected to construct these two mock communities. The above mock communities were analyzed using genomic analysis, and the preliminary results confirmed their quality. So here, I just want to say thank you for your attention, and thank you to all our collaborators.